Move to the introduction of aquaponics, and we're going to make an overview of aquaponics to be sure that everybody will get the same level at the end of this morning. So the goal we will have 3 goals: to present the different aquaponic system, to present the compartments of aquaponic system, and then to have a brief overview of the microorganisms, their location, and their potential roles. Okay? So let's move first to the presentation of different aquaponic systems. So when we speak about aquaponic system, it's a conjunction of 2 important systems, aquaculture and hydroponic. And of course, this ecosystem is believed to have less environmental impact due to these eco-services that each organism gives to the others. So we have a symbiotic relationship between fish, microorganisms, and plants. And you know that, of course, the fish sludge, fish faces mainly, are degraded by bacteria. The bacteria, and we will come back later on, the bacteria give some nutritive elements for the plant. The plants absorb these nutritive elements, purify the water, which come back to the fish. This is the general action of aquaponic system. If we look a little bit further in details, we have bacteria, central point, of course, because we are working here in microorganisms. So bacteria like Nitrosomas and Nitrobacter, which transform ammonia and ammonium in nitrite and nitrate. Okay? And the nitrates are the form of nitrogens which is absorbed by the plants. The plants transform carbon dioxide in oxygen, and at the same time, by absorbing, I told you, it's purified water. Water goes to the fish tank. Fish, when they grow, they transform oxygen in CO2 and produce sludge with nitrogen, but also orthophosphate. And of course, the rate of this nitrogen and phosphorus depends on the feed quality, on the composition of the feed, the state of the fish, and the species of the fish. So it's very a balance between all the microorganisms and organisms. And if you change one thing, like the feed, for example, it will have an impact on all the system. Okay? Then the solid particles are filtered out of the system. Very important. We will see why it's filtered out. And water is pumped to the biofilter back. And you've got this circle. So why hydroponic? Why do we use hydroponic? What kind of advantage do we have if we use hydroponic system? So hydroponic system is a soilless production. And if we have that kind of production inside the greenhouse, you can really increase the density of the planting. You will have more plants per square meter, higher yield. For example, if we speak about tomato, for example, in Holland, you can obtain until 80 kilo per square meter a year of tomatoes. In comparison with classical soil production of tomato, which is 20, 30 kilo per square meter. So you really increase the yield. You increase, of course, the production because you can work all around the year. Normally, you can have accurate control of the environment, including the root system. it means that you could control better the disease, the water quality, and the nutrition. And of course, because you are under a greenhouse system, you will have the control of climate, which is usable when you've got a high-value crop like vegetable, for example. Here you see why we could use hydroponic system in terms of environmental impact and in terms of use of water. So you see that in open field, you need 60 liters of water to give 1 kilo of tomato. 1 kilo of tomatoes. But otherwise you've got a big tomato. And you see that in parallel when you see from plastic tunnel, unheated glass greenhouse, and until even climate control glass, so warm and heated greenhouse, and of course with reused water, you obtain 15 liters per kilo. So you divided by 4 the use of water per kilo of tomatoes. So this is one of the main environmental advantage of using hydroponic. Why aquaponic? Aquaponic, of course, with the cycle and the transformation of the sludge into nutritive elements, you don't need, or you suppose not to need, to fertilize the plants. So you can see here, hydroponic tomato receive, in average, that kind of kilos of nutritive elements, so fertilizers, per hectare and per year. So you can see that if we can replace these fertilizers which come from chemical industry with or by biosource fertilizers, it will be another great environmental advantage of aquaponic system. So what are the major stakeholders in aquaponics? If you have questions, don't hesitate to ask me during the talk. So aquaponics, you've got, of course, the fish, the microorganism, water, plants, and also the sludge. I forgot here to mention this compartment. We often forget about water. Water is the main vehicle of all the system, and this is the main actor which link all the organisms together. So it's really an important actor. And if you look to an aquaponic system, so you've got the fish tanks with air pump to help to give oxygen to the system. From the fish tank, you've got a filter. It can be a mechanical or a settler. We will go back on that after. To discard solid sludge. Then with the pump, the water go to the biofilter, where you have your nitrifying bacteria. And then the water go to the grow bed, when you've got the plant growing on a substrate under natural light or under artificial lights. So this is the different compartments, the synthesis of these different compartments. Of course, and this is something we know, we use aquaponic system for professional use, but also for private use. And when we look at the private use, there's a lot of different ways to implement the system. And I want to show you 3 or 4 different ways for private use. I will also show you some a system for community use or urban community use and for professional use. So let's start with private use. You can see here a really simple, this is the simplest system you can find. You've got only one tank for the fish and one grow bed. Okay. Of course, it's a volume ratio 1:1 because you can't do something else. It's simple, but the problem of this is that you've got a variation of the level of water which can give stress to the fish. So we've got some other alternative, and they call them SHOP, like constant 8-1 pump. You've got all the time a pump coming from the sump tank to the fish tank, and here with this pipe you have a constant level of water for your fish, so no stressed fish here. The disadvantage is that you have to put your sump tank, and the shape of the sump tank is very depending on the height of the grow bed, where the grow beds are, and of course this sump tank must be able to contain water of both grow beds. So this is a constraint of that kind of system. It's more reliable, but you have a constraint for the sump tank. Another also way to control the height of the water here. This is SHOP 2. It's also simple and flexible, but here the water is pumped from the sump at the same time to the grow bed and to the fish tank. Okay? So here it means that the sump volume don't have to contain the equivalent of both volume of fish tank and the grow bed. This is an advantage. But the disadvantage is that you've got a reduced biological filter due to non-direct flow to the grow bed. Okay? And another example, last one for private use, because aquaponic systems are also more and more popular in tropical countries. Here they reuse sometimes barrel to make that kind of system, and so it's a very, very low budget system with one pump only, and the fish tank is just here. You've got your grow bed, and here the reserve for the water. So that was for private use. In the lab, we also try to develop some system. You can see this is really preliminary system, and maybe we will try to. The idea is to make some pilot and to see if we can have that kind of system for private use and integration in your lunch or in your kitchen or so on. And here, this is some existing systems that you can find in the market, and really small one, as you can see here. Oh, here it's already a little bit more complex with the aquarium and then the plant. I would say plant like aromatic plant, and also here tomato. So we've got also some example of system for urban community uses. And here you've got a system with a container and a greenhouse on the top of the container. And you can see that they are growing vertically, the plants. It's a system we will see with a kind of NFT technique, nutrient film techniques. I will come back later on that. And here, this is a German company located near Freiburg. And they are working on development of small greenhouse with, they are also looking at the energy. And they develop some solar panel that can be orientated depending on the sun and still give light to the plant and also absorbing energy for the system. And they develop that with an aquaponic system. We work also on urban community aquaponic system. This is our pilot, so I want to give you some idea of what was done in the PAFF box. So inside the container, we've got 2 fish tanks, the sump, a biofilter, I think, the pump and the biofilter, and the water goes up to the greenhouse on the top of the container. And the first year, in 2013, we had a ebb and flow with clay bits. So we use clay bits as a support for the plants, but also as a support for the biofilter, the bacteria. Here, the biofilter contains bacteria, but you can imagine that the water circulating in all the system can also take bacteria, and you've got bacteria also on the clay beads. Here you see how it was built. You will visit it tomorrow. And the first year was just to have some practice. You have to realize that we come from the plant world and not from the fish world. So we decided to use tilapia, which is convenient. I will tell you later on why it's convenient. And we're able to produce tilapia. And we tried different species, different plants, basils, lettuce, tomatoes, even this small pepper. So in 2014, we want to improve the system. And we compare ebb and flow with clay beads and rafting. So on the same system, we want to have a comparison of both. And you can see here, so the grow bed with the rafting system, and here's a grow bed with the clay beads. And you can see that we obtain some growth in both systems. I will come back later on the yield we had. And in 2015, we again update and upscaled the system. We put a filter because we had some problem with the solid sludge. So it was very important to discard, to take out this sludge. Then we decide to work only with raft system because we had better yield with raft system than clay beads. The greenhouse here, it's a really small greenhouse. So during the summer, if you have a lot of sun, it can create a microclimate with high temperature. So we decide also to have an air extractor. And to avoid also for that to have Vantomax, which is automatic opening of the window on the rooftop of the greenhouse. And then we also have a pilot unit for professional use. So we have a RAS system. You will visit it also. It's in the garage, we call it. So you've got here a special isolated room. And then you've got 3 tanks. You've got not anymore a decantation. Oh yes, you've got a decantation tank, but also a drum filter, biofilter, and you've got the sump. And here, why do we call it a pilot for professional use? It's because here we are working with a decoupled system. A decoupled system, it means that water will go to the greenhouse when we want. We open here or we close the pipe. So it means that if we close the pipe, it's just a RAS system, so recirculating aquaculture system. And we send, if we want, water to the greenhouse.
How do you determine the retention time of the water in the greenhouse then? Like a day or a week, or what do you base that on?
Here, for the moment, we just work as a laboratory. So it means that we take the water from the RAS and we go to the hydroponic system. We don't have a retention time calculated, but it's really important that that kind of calculation must be done. It will depend on the consumption of the nutrient by the plants. So you need to have sensors in your greenhouse to measure electroconductivity, and even more if you can measure each mineral constituent, then it's even better. You've got here an example with the urban farmers. And I'm sure Zala know better than I do. So it's in Basel. Hmm? It's in Basel, and the idea is with this container linked to a greenhouse, you can have a year 5 tons of vegetables and a little bit less than 1 ton of fish. So it's a professional aquaponics system, but some others are also developing around the world. So these urban farmers are now developing a new system in Den Haag. It's in Holland, in the Netherlands. Hmm? And you can see that they will have a rooftop of 1,500 square meter here. It's already finished. Yeah, it's already finished. It's finished end of March.
The grand opening is end of May.
End of May. But they say the farm is already working, and they say that it's working.
20th of May. 20th of May.
Yes, here 50 tons of vegetables and 20 fresh city fish they plan to produce a year. So it's already.
What is the fish species?
At the moment they have tilapia.
Tilapia. And they will produce what kind of vegetable?
Everything.
Everything. Oh, hydroponic.
Peppers, salads, tomatoes.
And where the fish are located?
So you have 2 floors. At the top you have.
At the top, okay.
Yeah.
No.
At the top you have a greenhouse, and 1 floor below you have the aquaculture.
So aquaculture is here.
Yes.
And you've got also another example here with the ECF farm system. It's in Berlin. theirs is not on a rooftop, it's on the soil. And they've got a greenhouse, and the aquaponic system is here. So with this system also, when you go to professional use, you can always see that the fish are under a covered system. It's not, they don't put the fish in the greenhouse. They separate the location of fish and plants, because fish needs, if it's tilapia particularly, it needs, the water needs to be heated, so they prefer to have it in a separate location when you've got isolation of the building. And here it's another example. It's in UK. The name of the company is Grow Up, and apparently they are making aquaponic system with NFT system with a multi-layer crop, because of course if you have a multi-layer you increase the yield per square meter. So just to give you some example of aquaponic system. Now let's move to the compartments. So when we speak about the different compartments, we've got plants, we've got water, which is going everywhere. We've got microorganisms. These microorganisms can be everywhere in the aquaponic system, even in the air. So it's very important to know it. Sludge, it's another compartment with, I told you it's important to discard sludge, solid sludge. And you've got the fish tank. So let's start first with the plant and grow bed. So here you've got a list of plants suitable for hydroponics. It's not exhaustive list, but it gives you a good idea of what can be grown with the hydroponic system. You've got leafy vegetable, you've got fruity vegetable, and you've got aromatic plants. And of course, in the industrial world, they don't produce bean or pea in hydroponic system. They still produce it in the soil, with soil. But for example, lettuce in Belgium, 75% of the lettuce are produced in hydroponic way. Tomato, you've got also a percentage with two numbers. So high percentage of tomato is also produced in hydroponic condition, also with pepper and cucumber. So it means also that hydroponic is very important in our world, and how important to produce major crops for the people. Just to remind you that if we want to give nutrition to the plant, we've got what we called essential elements. Essential elements are elements which are required for the plant to make the entire cycle from the seed to the seed. And these elements are not, we cannot replace this element by other. This is why they are really essential. And we've got 16 elements that are essential for the plant. And if we look at it, we've got, of course, hydrogen, carbon, and oxygen, which are coming from water, from CO2, and from oxygen and water. And you can see that they can be in high concentration in the tissue of the plant. You've got nitrogen, and this is the available source of nitrogen for the plant. And in average, this is the concentration in the tissue. you've got potassium, you've got calcium, phosphorus, it's with the orthophosphate form, sulfur and calcium and so on. So these are the concentration, and the less concentrate here it's sulfur. And when you go to the micro or micro elements, this is the 8 micro elements necessary for essential for the plants. And you can see here that the concentration in the tissue is far away, lower than the macro elements. Okay. So it's important to remind that when we are speaking about aquaponics, because maybe in the sludge you won't find everything. So we have to make sure that we have at least all these elements. And we observe, for example, in our system that iron can be a problem. I mean, we are missing often iron in the system, in the water. And sometimes we have to spray chelate of iron to be sure that we don't have chlorosis of the plant. Important point also is the optimal pH for the plants, which might be not all the time be the same than the optimal pH for the fish or even for the bacteria. So this is the bacteria who are nitrifying the sludge. So it's very important to remind that also. And if we go to a soil crop, soil culture, this is the optimal pH range from 6.5 to 7. If we go to hydroponic culture, the optimal conditions or the range is between 5.5 and 6.3. So it's a little bit lower with hydroponic system. And also, this is important to remind that. Other factors that are influencing the plant nutrition are all the different factors that will affect the plant growth. So it means plant species, plant stage, temperature, light, relative humidity, and also the substrates. Substrates, are you familiar with the substrates? What kind of?
Turf. We use turf, although it's soilless.
Turf.
Compromise between like something inert and something that is
Yes.
naturally
Other substrates than stone wool, cocoa fiber, or turf? Perlite, vermiculite, the clay. Okay. So these are the different systems for plant growth. Again, just to remind you. So we've got drip system. So a drip system means that you've got drip on each. You irrigate the water on each plant. Okay, and water coming back from this plant to the tank, the sump. NFT, you have just one flow of water, so you don't have water which is given to each plant, but just one general source of water. This is a difference with the drip system. And we call it nutrient film technique because you've got a real film beneath the plants. And also you really need to have here in pond. A slope of between 1 and 4%. And from the slope, you will have a drain tube going back to the reservoir and start again. So here the system, the first system, the drip system, is, it's for example, big plant like tomato, like pepper, because they can fix their roots on stone wool or fiber, cocoa fiber, but big system. And with NFT, you will not be able to use such a big crops, but you will use, for example, lettuce, aromatic plants. It's more usable or suitable for these plants. Another advantage of NFT, because you have a nutrient film, it's very light. So one can imagine to put that kind of system on a rooftop. Remember that for a rooftop, if we want to go to crop on a rooftop, one of the most important constraints is the density of your crop per square meter. If you come with soil, I mean, it's really heavy. And here, it's just, of course, the weight of the plant and the weight of the nutrient film. An NFT system is really well known, really it's used in a lot of different countries, and you can see here it's used, for example, to produce aromatic plants or lettuce. We also have vertical system, as I told you. And here it's with strawberries. And these systems, when you see, they are shaped in a way that the plant, each plant can enjoy similar light and similar quantity and quality of light. You've got on NFT also what we call A-frame. And you can see here the system. And you've got different pipe which supports the plants. And these different pipes, again, are made in a way that the plants, all the plants have enough light and distribution of light is equal. You've got some, I would say, private system also, cascade, we call it, as you can see here. Then if we come to the deep water system or raft system. So this system is with water. It's 15, 20 centimeter. So the advantage is that you have higher volume of water. So it's the buffer, if you have a problem, is higher than NFT. But the disadvantage is you use more water at the beginning, and it's heavier than NFT. Okay. This system, for sure, you have to take care about providing oxygen for the roots. And sometimes you adjust the pipe to get rid of the water a little bit lower to be sure that part of the roots will have contact with air, direct contact with air. As you can see here, the pipe to evacuate the water is a little bit lower than the raft system, so part of the root system will have contact with the air. So it can be important. Ebb and flow system. So it's ebb and flow system. You have here a pump which sends the water to the grow bed, and after a certain time the pump stops and the water goes back to the reservoir, okay, when the pump is off. So it's also a system which is often used, and I would say that you can use it with hydroponic system, but also with normal soil system. You can replace inert substrate with soil, and it's working also in a very good way. Finally, the last system for plant growth is aeroponic system. Aeroponic system is a system with sprinklers. So the advantage of this system is very light again, because you don't use a lot of water. The second advantage is you save water. And the disadvantage, it's the fragility of the system. You can imagine that if your pump stops, your roots immediately start to dry. So this is really a fragile system. So we need to have a second pump, or to have a kind of alarm system to say, okay, there is no electricity anymore. Another weak point of the system is that it's sprinklers, and they can be, you can have some problem in the sprinkler. And of course, with aquaponic system, if you have some, you can have some debris, you can have some solid sludge which escape to the filtration, and then you will have problem with your filter. So another point is that with aeroponic system, you really stimulate the growth of the roots. And in some case, you have a lot of roots, but you don't have a lot of leaf. So it's still, from one article to another, the crop yield is still controversial. I don't know if you had some experience with aeroponics. None of you?
Once, yeah.
Once?
Yes, it was a huge amount of roots, not that much.
So you had similar results as us, and Kevin?
So we got a very small system, the same, only the roots.
Mm-hmm. I was in contact with Italian company working on the development of aeroponic systems. They told me that they had really good results in terms of yield. And they say that their market is countries where you don't have a lot of water, like Turkey, for example. Or we had some experience in our lab 3 years ago, and we obtained good yield with other systems, but not with aeroponic. We didn't have a lot of yield. And here, this is a comparison done in the PAFF BOX. I told you that in 2014 we compare rafting system and clay bead system for lettuce and for basils, and you can see that at the same stage. We start here mid of April, and you can see that weeks after weeks, the difference are really huge between deep water rafting system and ebb and flow. And we obtain similar difference with lettuce. So this is why we decide to carry on with rafting system. Again, do you have experience of comparison of clay beads with rafting? And
Yeah, we compared NFT to gravel and raft. We didn't find any difference between raft and gravel or even flush systems. Although NFT performed only 50% as good.
NFT?
NFT was only half the production compared to the other two.
Okay.
Very low. I don't know if it's the quality of the system itself or what might have been the reason. All the other factors were the same: water, nutrient addition, everything.
The flow. What was the speed of the flow? Similar or?
Approximately. Just for one NFT pipe, maybe 1 to 2 liters every 5 seconds, 1 to 5 seconds, approximately.
company in Canada that I went to, and they use the NFT with a pipe system, but it's 5 centimeters of water.
5 and not 1, okay.
Yeah, and it's half and half. And he had the raft system, and he stopped using it. And now he just uses a 100 centimeter pipe, 100 millimeter pipe with 50 millimeters water, and he's got air, water flow, and it's perfect.
It's perfect.
3 years he's been working that system.
Okay.
You don't have the weight of the water, you don't have the
Is it
expense of the polystyrene beds.
this is a system of Nick Zavidov?
Yes, one
Okay.
it's from Nick's, the generation 4. He uses that, and he put the water level up on that, and he's a perfect result. Some beautiful, nice clean roots, and water temperature stays the same. It's good for tropical conditions also, because you don't have your NFT heating system problem.
Yeah, because of this
You
sort
put
of...
your water level, and if something happens, it doesn't dry out. You've still got your air to the roots and your water temperature.
Okay. If we go to a second compartment, this is a fish tank, and I have to tell you that we are not familiar with fish, because our laboratory, it's a plant pathology, so we are familiar with plants. So when we look at the literature and we see what kind of fish are easy to handle and resistant to our mistakes, I would say, and we selected tilapia. For us, tilapia was the choice because it's able to, indeed, it originates to African lakes and from warm water, yes, but is able to grow with a lot of tolerance in terms of pH, in terms also of oxygen dissolved, temperature and ammonia and nitrate content. So for us, it was an advantage to start with tilapia. You see that tilapia is, the name is the Latin name. And we also choose tilapia because of the diet of tilapia. It's omnivorous. So it means that it doesn't need to eat a lot of proteins. 30% of proteins is necessary for him, for this fish. And then we search, or Boris searched for a feed which contain only vegetable and plants, and we find one. I don't remember where does it come from. Aqua4C, okay. And so the standard diet is only sourced from plants and contains 30% of proteins. I'm not sure we won't be able to do the same if it was trout, for example, which is carnivorous. I don't know, we can also make a... What kind of fish do you use, people who are working with aquaponic system?
Carp.
Carp? Carpus?
European catfish.
Catfish?
Can you feed it only on a veggie-based feed?
Not yet.
Not yet?
Not yet.
Trout?
I get some industrial food for now, but I'd like to go with the insects.
Insect, yeah.
They made
That's a new...
research in the university near my town. They made a research about the black soldier fly. It's called black soldier fly larvae.
Yeah, for the larvae, yeah.
I'd like to try those guys, but now they are still studying if they could bring disease to the fish, so now it's not clear now. Well, I will try within the next months. I will see.
Okay, Kevin?
We were growing trout and also carp and largemouth bass. That's an American fish. No tilapia because I don't like to heat the water in the summer.
Yeah. Okay.
Small scale also have trout, tilapia. Mostly just carp and trout.
More or less tilapia, otherwise also burbot, timulus, pike perch, sturgeons. So we tested a few of them, but for now we stick with tilapia.
Okay.
Tilapia also, sturgeon.
Tilapia
Sturgeon.
and sturgeon. Okay. Other.
Tilapia, African catfish, and I'm involved in different projects with PECA communities.
Perca. Perch. Perch.
European perch.
And the sturgeon can be vegetarian also? Can you feed it as vegetarian dish? No, they need trout.
So now I have a problem with my computer.
In the place that we see from far, Thursday, you can see they grow another fish. It's called jade perch from Australia. And this is more easy to grow on just on veggie base.
And here you can find some fish species. We speak about carp, we speak about trout. You can see that the carp are also omnivorous. So. They've got good tolerance to different conditions. Trout, of course, you've got a market in Belgium, you've got a big market for trout, but it's is it an advantage or not? It needs high oxygen concentration, it's less tolerant. Perca, it's growing fast, good adaptation and tolerance. It's carnivorous, and it's, I would say, cold or water. And of course, we often forgot that we can have also some other fish. We don't eat them, and we have to think about these fish when we speak about private use. Because if at home, I'm not sure that people will like to kill their fish. You can imagine the small children, oh, it's a nice fish, and so we have also to think about this if we speak about development of aquaponics system for private use. Okay. So if we go to the compartment of sludge or solid, you can see that there are a lot of different ways to get rid of the solid sludge. It can be incorporated directly to the fish tank, like here. You've got an incorporated settler here. It can be a separate settler. It can be a more sophisticated with different chambers. We can use some drum filters. You need electricity and energy for that, or also sand filters. But I'm sure that Amit will present more this system on Wednesday. So water, we've got water everywhere. And quality of water is very important. Again, Amit will speak about quality of water and how to measure it. But I would like to present some results we obtained in the PAFF box, and it will help you already to see what kind of parameters reflect the quality of water. So here we survey the quality of water in the PAFF box in 2014, and you can see the average of temperature. So it's part of the quality of water, of course, because remember that some plants need lower temperature, some plants 24 it's okay, For lettuce, for example, it's better lower temperature for the roots. You've got the pH, average pH during all the period. The electroconductivity. Are you familiar with electroconductivity, all of you? It's reflecting the concentration of the ions in the solution, so it reflects the nutritive concentration, nutritive element concentration. We had a high level of electroconductivity, the demand of oxygen, and the tan. And the tan, you know what is the tan?
Total ammonia nitrogen. Total ammonia nitrogen.
So it's a both form of ammonia that you can find in the water. And it's of course really important because the tan can be, if it's too high, it can be toxic for the fish. So we compare what we obtain in the PAFF box with what was obtained in the literature for hydroponic system and for aquaponic system, you know, Rakosi. I would say that this is a father of the modern aquaponic system. And it was building a system, a big system in Virgin Island. So it's more a tropical system. But anyway, this is why we choose also to check with the number from Rakosi system. And you can see that in that period, we had quite lower concentration of NPK in comparison with the standard hydroponic system. And I just remind you that NPK is three major macro elements for the growth of the plant. That time we had a lot of calcium, but it's because we use top water, isn't it? Yes, huh? So that here we use top water, and in Gembloux you've got a lot of calcium in the water to adjust the system. So when we look at the quality of the water, here we also look at the different level of nutritive element or mineral elements. And you can see that, for example, for the nitrogen in nitrate, we've got a small increase through the period of 4 months. Okay. You can see that for iron. I told you that we had sometimes problem of iron. So with our method to detect iron, we were not able to detect iron only. So it can be a real problem for. Ooh, good question. Detection for iron, do you remember? Detection limit.
It's I don't know exactly.
We can have a look later on.
It's with a spectrophotometer, so it's not very high precision.
So if we look at the relation between the dynamic of the different nutritive elements or mineral elements and the plant yield, you can see that we obtain similar yield. So we grow two varieties of lettuce. Grosse blonde paresseuse is a lazy fat blonde variety. This is the name. But it's a variety which is really fragile. And we want to show that we are able with aquaponic system to grow that kind of lettuce. And that kind of lettuce can be very appropriate in an urban environment where you just harvest and eat, and you don't need to transport the lettuce everywhere in the supermarket. Sucrine is more a marketable variety, or already well known. And we didn't have big difference between one period and another. And we had quite similar yield in comparison with what can be obtained in hydroponic system, despite the fact that I told you phosphorus, nitrogen, and calcium and potassium were really low in comparison with hydroponic system. With tilapia, we also see what we could obtain for tilapia. And you can see that we obtain an increase in 4 months of, let's say, around 17 kilos. And you see the average weight from the beginning and at the end. And few important, this is a conversion ratio. In literature, it is said that it's near 1. We had 1.3, so it's not too bad. And they were resistant to us, 5% of mortality, and the grow rate was 1.97 or 2 grams a day. Water consumption, because it's important also to see. You can see that the total research water use was more or less 8,000 liters for 4 months. We had to fill up the system with 2,000 additional liters. And the average daily water research was 60 liters. Okay.
This was in the PAFF box.
In the PAFF box, yes. It's result from the PAFF box. So again, in the PAFF box, 1 year later, in 2015, you can see here that the macronutrient, so average temperature, pH were nearly similar, but the macronutrient was lower than in 2014. We observe at the same time a decrease of potassium, magnesium, and calcium in the water, maybe due to the fact that we use rainwater that year to fill in the PAFF box. But despite, you can see here the dynamic of the different nutritive elements. You can see also that here we stop the experiment for a few weeks. Sometimes Boris has to have some holidays, it's normal. and we start again with Boris and James, STSM student. And you can see here, it's very clear that we have an increase over time of nitrogen, for example. But again, it was, as you can see, it was lower than the optimal concentration for hydroponic system. And despite this lower concentration, we could obtain again, during several cycles of lettuce grow, good yield, except the third and the sixth. I don't remember now why.
Those substrates. That was on other substrate.
Ah, yes.
You used cocoa beans.
Normally here you use cocoa peats there. And here it was the end of the season. I think it was already colder.
Less light also.
Yes?
About the potassium.
system or?
No,
Because I see the graphics.
here it goes in such a way.
I've not really explanations on water quality.
It might be, yes.
We exchange some water at that point between the two parts. I mean, before June, at the end of June, we exchange some water. Yeah. Because the idea was we didn't want to add any nutrients instead of just the feed, and we just added rainwater and tap water. And to control the pH, we just exchange water. So we wanted to see what's happening when you don't add any nutrients.
Alkalinity in the water? Yeah, tap water.
This is also,
We
yeah.
control it with the tap water. So we know exactly which amount of nutrients that were in the tap water.
But here it was with rainwater, and to solve, yes, to regulate the pH.
We use tap water.
Tap water, okay.
Which is hard. We have a lot of calcium and magnesium here in the water.
Do you know how high is the alkalinity?
Of the tap water, it's that's also a question. It's around 80.
And here you see the concentration of the micronutrients inside the.
It's 80 milligrams per calcium carbonate? Low.
And here you see the concentration of microelements, micronutrients, and you can see here the difference. And you can see also that for iron, for example, we had some iron. I think it's because we had some.
No, we didn't add it. It's because we use a more precise method to
That was a new method, a method with a limit detection lower.
But it's in milligram, in
Microgram, huh? Yeah.
So for all this we use another method, not photometer.
It's total ferrous or dissolved?
Sorry?
It's total or dissolved? With the Fe3 and Fe2, only Fe2. Did you filter the sample before?
Yes.
It's only Fe2.
It's total.
No total, only dissolved.
Yeah, dissolved in the water.
So here you've got the production. So it's of course higher production because we run the system for more months. So more or less 30 kilos of additional kilo of tilapia. And you can see that the ratio of conversion is a little bit higher in 2015 in comparison with 2014. And mortality was similar, but the growth rate was also lower in 2015. Yes?
How were the fish fed?
Continuously.
We changed, so between 2014 and 15, we changed the feed. Here it was the normal standard feed with also animal proteins, and here it was feed only with plant proteins.
But they were fed by hand, automatically?
Yes, by hand. No, they were the thing is we decided to have a good view of the water quality evolution. We decided to give every day the same amount of feed. So at the end of the experiment, they were a little bit underfed because they grew up. But that was the idea to have a good view of the nutrients.
So same amount every day, not at libidom.
Did you feed them or with the samples between the feeding, you know, or you just took the samples when you could, because you
No,
can't
no,
make
it's
bits
every day the
yeah,
same
after the
time.
feeding.
Yes.
Same time of the day. I don't remember when exactly that was. But I mean, only for nitrogen you will see changes.
Also the phosphorus, but also
So in the water consumption, we have higher water consumption, but of course we run the system need double number of months, so it's a little bit normal. But daily water exchange, so liter per kilo of feed was lower in 2015 in comparison with 2014. So we had slight difference for tilapia rearing performance, and that could be explained by the feed composition. We had difference in water quality in terms of electroconductivity and also in macro element from one year to another. But however, no slight difference for lettuce growth. So that was really surprising us. We have a good growth of lettuce. And so we had a development of plants with reasonable yield despite low concentration in nutritive elements, whatever the year. So that was an important result, and from this result we start another experiment that I will explain you later on. But just remind that with aquaponic water here we had good yield like expected for hydroponic crop.
Your FCR, your smaller fish the second year, 73 grams.
Yes.
63 grams.
Yes.
To explain your FCR.
The FCR, yeah.
Fish
Yeah.
For growth FCR, unless it gets bigger,
Exactly.
the FCR will drop.
Yeah, I forgot to explain why here, but you're right.
Professor, did you measure the nutrient content in the plants, in the biomass of the plants also, compared to typical hydroponic systems?
This is ongoing results.
Yeah.
Okay.
We did it, but not for. Yes, we have this, but it's very processing.
Yes.
Okay, because we are just processing this kind of research right now with the question, and in fact the background question is how is the uptake of nutrients by the plants in an aquaponic system due to all these microbiota that we have,
Mm-hmm.
and compared to a classic or standard hydroponic system?
Yes.
When we get the results.
This is something we're also working on, of course.
Okay.
So let's move now to the last point for this morning: location and potential role of microorganisms in aquaponics. So it will be a brief overview before this afternoon and tomorrow and the day after. So I prepared this with the help of Gilles and Yasmina, who she was a master student 2 years ago in our lab. So, microorganism. Where can we find a microorganism? I told you we can find it everywhere in the system, in the air, and you have to realize that in the air, in the winter here, we have less microorganisms in the air than in the summer. So already there, you can have an influence of the air to the aquaponic system. And for the aquaponic system, it can be everywhere in the water, plants, fish, sludge, the system itself in the pipe, the biofilter. So microorganisms can be everywhere. And if we look at aquaculture and RAS literature, this work was done by Gilles, we can see that the density of the microorganism and the diversity of this microorganism depends, of course, of the species of fish, the type of water, and what kind of feed you give to the fish. It depends also on the O2 and nutrient gradient, on the other environmental conditions like temperature, like C/N ratio. This is very important. I will come later on this C/N ratio, pH, and so on. It depends also on the filtration system, how you can get rid of the solid and so influence the C/N ratio, and the biofiltration. And also, it depends on how you manage the system in terms of waste, on disinfection, and so on. When we speak about microbial classification in RAS system, in aquaculture, but also in aquaponics, we classify them depending on the mode of nutrition and on the metabolism of respiration. So you can have strict aerobic, facultative, anaerobic, or optional or facultative anaerobic. And the mode of nutrition is autotrophic and heterotrophic. So who can explain what is the difference between autotrophic and heterotrophic?
Well, heterotrophic organisms receive their energy by the oxidation of preformed organic molecules, compared to autotrophs who take energy, and especially carbon is interesting from the air. So, well, you have to add photoautotrophic, then you also know where the energy comes from. But basically, they take the carbon from the heterotrophs, and heterotrophics take it from oxidation of preformed carbon molecules.
Do you agree about this definition?
It's actually about the carbon source. Yes, autotrophic take carbon from CO2. So nitrification is their plants, and heterotrophic take carbon from vapors.
Yes, exactly. So, of course, heterotrophic bacteria are able to take the energy from carbon.
Oh, the heterotrophic autotrophic refers to carbon source, of course.
The carbon source. So, if you look at the fish, of course, microbial population allows the processing of the waste from fish. And of course, it is one of the most important study and well-known microorganisms in aquaculture or in aquaponic system are the microorganisms able to convert nitrogen in nitrate, so the action of nitrification. And this is done by autotrophic bacteria. So it means that they are not using carbon from organic matter, but from CO2. Okay? And they are also working on other conversion like proteolysis, like sulfate reduction. And when we speak about degradation of organic matter or solid sludge, then we are speaking about heterotrophic bacteria. This is a major difference. So when we have a look at the group of bacteria in fresh water, and I told you in literature this is well known, but for example, we don't have a lot of study on fungi, which are also microorganisms. We have really a lot of literature in bacteria helping to transform and helping to the nitrogen cycle. There we have a lot of knowledge. And here we find the different groups of bacteria. You can see that it's actinobacteria, that proteobacteria, but also bacilli, nitrospirae, planktomycete, and also Nitrosomonas. We will come back on this. So for nitrification in fresh water, you've got Nitrosomona, which is really well known, and Nitrospira. But we have also other genus that can help to the nitrification, which are Nitrosococcus, Nitrosospira, and for the second step, it's Nitrobacter. So when we speak about heterotrophic bacteria, as I told you, they can be in competition with autotroph for oxygen and space. So this is why it is so important to discard the solid matter from your system. Otherwise, heterotrophic bacteria will take advantage, and you will not have a good development of the autotrophic, which are able to transform ammonia in nitrate and nitrate.
Heterotrophic outcompete?
Yes, exactly. So they degrade organic matter. They're also able to produce subproducts. And they also occupy several ecological niches. And on the other way, there are also these heterotrophic bacteria. For fish themselves, it can be really important because they can limit the chance of proliferation of opportunistic microbes. So you've got advantage and disadvantage. If you are speaking about RAS system and fish or aquaculture, there are advantages to have this kind of microorganisms. You want to add something, Jochen? So, and this is why it is so important to, I told you, to have, to get rid of the solid sludge. Here you see some representation of the biofilter. I told you that in some system you've got bacteria, nitrifying bacteria everywhere. And if you have clay beads, for example, you can have, this is a material that can support and stimulate the growth of the nitrifying bacteria. But in some cases, you just don't have the clay bead. You just have, as I told you, NFT or deep water, and then you need a proper biofilter. So it can be with that kind of biofilter with some lamina, which is able to, what is important for your biofilter is that the surface, in comparison with the volume occupied by your material, the surface is very high. So you have a really high surface in comparison with the volume. So it can be with lamina, with this small, you can see here, small plastic, just gonna, few seconds, switch off. You see the small plastic here. Again, you've got a lot of surface for the volume. You can use that kind of system which contain these plastic beads. So biofilter is important for nitrifying system. And here you find nitrifying system or nitrifying classification. So we've got two important parts when we speak about nitrifying. We've got the ammonia oxidizing bacteria and then the nitrate oxidizing bacteria. So from ammonia to nitrate, it's a two-step process with AOB and NOB. And you've got here a classification of the different AOB and NOB, and you can see that we find back the Nitrosomona, the Nitrobacter. This is for the second step of the process, for example. We see the Nitrospira, as I told you already, the Nitrococcus, and so on. So you have here the classification of the different bacteria for the nitrifying process. So, I just told you that, and these are gram-negative aerobic. It's important to say that it's aerobic bacteria and chemolitho-eutrophic. So we see also that nitrification or nitrifying can be realized by other organisms like fungi, like heterotrophic bacteria. And if you see the reaction, of course, the interest for the bacteria, the autotrophic bacteria, is to get energy. So it means that the chemical reaction must give energy to the bacteria. And you can see it with the ammonia. And this two-step reaction give you nitrate and give you, during 2 times, energy to the bacteria. And then with the second step, the NOB, so nitrate oxidizing bacteria. Here, the reaction, it's also a 2-step, but you have to notice that the first step is not giving energy to the bacteria. It takes energy to the bacteria, but the second step gives energy. So at the end, the balance, the total balance, means that the bacteria will receive energy by doing this chemical equation. Another important point, and this is important to realize when we are in an aquaponic system, because nitrification, you can have optimal conditions and suboptimal or non-optimal conditions. And when you see with both AOB and OB bacteria, the optimal condition is around a pH 8. Remember the optimal pH for plants. It was lower. So in the same system, if you are working with one loop, you have to find a compromise for the plant, for the fish, and for the bacteria. Okay? Again, also for the temperature. Temperature, you can see the optimal activity are in higher temperature, 30, 35, 30. So 30, maybe it's good for tilapia. It's not so good for the plants. So again, we have to find a compromise between all the requirements of the different organisms. And here it's a graph showing you why it is so important to discard solid particles. When you see here, this is the ratio C and N, and when you've got a ratio where carbon is higher than nitrogen, you decrease the nitrification rate. Okay? So this is why it is so important. And here you've got a table. It is in French, but I'm sure that pH, O2, O2 limit, temperature, so it's okay to read this. So bacteria, tilapia, and plants. And you can see that when we speak about ecological niche for bacteria, it's more close to tilapia niche than plant ecological niche. Okay? So I don't know if you are familiar with couple and decouple system. Who can explain what is a decoupled system?
The coupled system is a system that you can separate the plant production from the fish production. In order to, if you have to do a treatment in plants, and couple, can cut the separating system.
Boris, what is your explanation of decouple and couple?
For me, it's you have an aquaculture system working in one loop and a hydroponic system working in one loop, and you can at some point connect both systems. But usually in this system, the water goes only from the aquaculture to the hydroponic. So
Yes.
you go back, and the rate, the flow rate in between aquaculture to hydroponic is dependent mostly of the transpiration rate of the hydroponic.
So what is really important in terms of decouple is that you've got two cycles, but it goes from aquaculture to hydroponic, but don't go back from hydroponic to the, this is a decoupled system. And maybe there is some sense also to speak about decouple when you see this. And when you see that, and when you have decoupled system, normally your biofilter is along with the fish system, the aquaculture system. And when you see that, it makes sense, okay? So if we look at the other part of the system, and still concerning the microorganisms. So where can we find microorganisms in the plant grow bed? Everywhere, as I told you, in the substrate, in the pipe surface, in the nutritive solution, in the rhizosphere. And again, the density and diversity depends on different things, different parameters like the type of substrate, organic versus inorganic. You might have less diversity and quantity in inert system. About nutrient solution, it depends also on the species and variety and the age of the plants. The type of irrigation, we saw NFT and so on and so on and so on. And also the environmental conditions, temperature, pH, and etc. We don't have so many articles in literature. We don't have so many articles. So when we speak about the main pathogens in hydroponic culture, we have fungal root pathogens with zoospores. It belongs to the Oomycota classification, and the main fungal root pathogens are Pythium and Phytophthora, and I'm sure Peter will speak about it. If we speak about other fungal root pathogens, this is a major one, but we have also fungal foliar pathogens, like powdery mildew, botrytis, and this is other one. So you will have more details on that kind of pathogens later on this afternoon. It's just to give you an overview, as I told you. So you can have pathogens, and you can have also beneficial microorganisms. And these are coming from a recent article last year, making kind of overview of what kind of beneficial microorganisms we can observe in hydroponic system. And you can see that we have a lot of bacteria belonging to the genus of Pseudomonas, of Bacillus, Enterobacter, and Streptomyces. And here you've got two beneficial that we can find which are belonging to the fungal, to fungi. It's Gliocladium and Trichoderma. And I will come back later on with Gliocladium as an example of biocontrol tomorrow. So for the plants, we have pathogens, but we have also beneficial microflora. And you can see that there are a lot of different species. And we can find them on different hydroponic plants, or plants that can be grown on hydroponics. And Gilles has made a review of what kind of microorganisms we can find in tomato, for example, and 40% belong to the Pseudomonas genus. So Pseudomonas seems to be very important if we speak about beneficial for tomato. So these microorganisms, as you have seen, has already been used in agriculture as biofertilizer, as biocontrol agent. Some of them are what we call plant growth promoting rhizobacteria. What does it mean, plant growth promoting rhizobacteria? It means that they, of course, promote plant growth thanks to the production of plant hormones, for example, but also fixation of nitrogen, control of plant stress, and extracting nutrient from soil or nutrient from water. But another point which makes them important also is that they are able by themselves to compete with the pathogens. We will see that tomorrow, and also to have some biocontrol properties. Okay. We also observe in hydroponic system suppressive substrate. What does it mean? So I don't know if you're familiar with suppressive soil. It's we can observe it was already observed, I would say 40 years ago, first in France in Avignon soil, and they could observe that if you take a soil, it's able to some soil, some plots are free of fusarium from certain crops. And when you take the soil and you put the soil in another one, this one acquires that kind of suppressive disease properties. But if you sterilize the soil, you put on the soil with other properties, it's still, the plants are still sensitive to the fusarium. So it means that it's so they conclude that it's coming from microorganisms. And they made exactly more recently similar experience with inert substrate from hydroponic system. And they could see that some of the substrate have that kind of suppressive properties. And if you sterilize the substrate and go back to your system, your plant are not anymore, your cucumber here are not anymore resistant to pythium, which is an important disease you will see this afternoon. Okay. So in contrast, the application of single introduction antagonist, in such experience, they try to introduce one microorganism and not a natural population. And with this one microorganism, they didn't succeed to have a protection of the pythium. So maybe it's important to deal with population of different microorganisms, so a real microbiota, and not to deal with one super strain that you can implicate in your system. Okay. And if we look at the literature in aquaponic, and I will end with this, you don't find a lot. Yeah, so for the moment, if we speak about characterization of the microflora in aquaponic system and their properties, potential beneficial, neutral, or pathogen, we don't have a lot of data. Even we don't have data, not enough. So we need to produce that kind of data. So I come back with the experience we made. So I told you from the PAFF BOX, if we compare aquaponic quality of water, we had really lower quantity of NPK in comparison with hydroponic, but still have what we think good yield. So we want to start in a real scientific control manner. So we took this kind of system that was done with Boris. We took aerofloat system and we put aquaponic system or hydroponic solution. So we put the optimal concentration that was determined a lot of years ago by Resch. And a third aeroflow we occupy with water we call complemented aquaponics. It means that we put water from the fish, aquaculture water, plus mineral salt to reach same Resch values. And then we compare the system. And you can see that in terms of, it was done on a variety of lettuce. And you can see that we obtain with complemented aquaponic water an increase of 39% of weight in comparison with aquaponic alone, in comparison with, hydroponic system. Okay. And in terms of yield, comparing aquaponic water and hydroponic water, we let a little bit lower yield, but not significant in comparison with hydroponic. So it confirms what we obtained in the PAFF BOX, more or less similar, maybe a little bit less when we have aquaponic. But what was surprising us, of course, is that if we complement, we obtain a big higher yield. And another point which is important to mention here is that concerning the root fresh weight, we had a high development of root when we have aquaponic water. So aquaponic alone or complemented aquaponic, okay? In comparison with hydroponic. But when you make the ratio, yes?
Did you compare the fresh matter and the dry matter of the green material, in fact, from aquaponic to hydroponic?
We have not enough data on the dry to be able to really have a good statistical analysis.
Yeah. So we haven't any statistic yet too, but the tendency is that aquaponic has lower dry matter than hydroponic.
Yeah.
Lower,
lower
dry matter.
But yet a higher fresh matter.
Okay. Good point. Interesting. So yes, we have 39% more yield with complemented aquaponics in comparison with hydroponics. So how to explain such results? We have two hypotheses. Maybe you have more, but we have two, which is, of course, we are working with aquaponic water, and there are some organic matter still, soluble organic matter in the water. So maybe it's due to the organic acids. But we also suspect that the microbiota might play an important role, and that the quality of the water, the composition of the water might influence the microbiota. And Gilles will work on that. He's working already on that. So to conclude, role of microorganisms, to be really simple, you can have plant and fish diseases, but you can have also beneficial roles of the microorganisms in plants it can help the nutrition with a biofilter, but also with some specific plant promoting rhizobacteria in the rhizosphere. It can help to protect the plants against disease and to enhance plant growth. And of course, there is also some roles in the sludge mineralization. And why we decide to make such a week of few days of training is because, yes, we believe that microorganisms play a really key role in the aquaponic system with bad or good effect. And really, if we understand better, we believe that we will be able to enhance the performance of the aquaponic system. And then I will end my talk with this question. Now that you have a general overview of the different system, compartments, and also the stakeholders, plant, fish, microorganisms, what do you think is preferable? To work with a coupled system or a decoupled system? It's a polemic question, so I think we have no time to go to eat and to discuss about that, and we will speak later on about, we don't need to answer today. Thank you.